I don't really need this one, so I don't think I'm going to worry about downloading it. But congratulations, our little mosquitoes. All right. Chapter 4, and this one's not as long, so that should be some consolation, I guess, is the word I'm looking for. This one's on infection prevention. Wrong piece. Okay, so here's some of the terminology that's important for this. Infection prevention, the chain of infection, I've added in a diagram that shows this: vegetative organisms, spores, and denatured are some other key terms, which are really seemingly random, but they'll make sense when we start to go through this. So this is the chain of infection, and it talks about the major links in the chain, which are representing, these are the things that we need to keep intact. And then each one of these little breaks are where you can have breaches in the armor or breaks in the system. And so you want to just be aware of all of these different components that are related to each one of these. So the first one here is the causative agent. This is rapidly recognizing if somebody has an infection and correct identification of the organism so that you can treat it more effectively. Source of the reservoir, where is this infection coming from? Is it through some type of contamination? Is it that one of the employees is sick and bringing it in? Is the disinfection not being carried out correctly? So these are the different areas where a breach in the armor could occur, basically. Portal of exit is just where the organism is coming from when it is leaving the host. Mode of transmission, how is it being transmitted? Are we keeping whatever part of how it's transmitted clean so that we can prevent the transmission? Portal of entry, again, is knowing where the pathogen is entering into the individual. So this can be different in different scenarios, different depending on what the patient's treatment is. And then susceptible host, kind of like we're doing with COVID, recognizing high-risk patients, if they're immunized, things like that. So I thought that was a really good diagram that kind of summarizes the whole thing and knowing all the different areas where you need to be aware of or be careful about. All right, we talked about the fact that some bacteria form endospores. And remember that an endospore is a dormant state of the bacteria. So it's basically, you can kind of think of it like it's going into hibernation because it's in a harsh environment and it wants to not get destroyed, not get killed. So when it is back in a nice, moist, warm environment, it can return to a vegetative state. The vegetative cycle, unlike what it sounds like, is the normal actively metabolizing, actively replicating bacteria. So if it leaves the endospore state and returns to the vegetative state, it is now replicating by binary fission again, and it is a live, active, replicating bacterial cell. This is the chart from your book that just kind of summarizes these different microbes and how susceptible they are to different levels of disinfectants. So this is a good one to just kind of know the relative. I have a more simple one later, but you can see that endospores are some of the most resistant because at intermediate and low levels of disinfectants, they are not killed. Vegetative cells, again, those are the normal actively replicating cells, are more susceptible. And then certain bacteria that are more resistant, for example, Mycobacterium tuberculosis that forms a capsule, is not going to be killed at low levels, but it may be at intermediate and high. Fungi can also be more resistant, as only some are killed at low levels. And then your naked small viruses are going to be more resistant to disinfectants, whereas your enveloped medium size viruses are going to be more susceptible to being killed, which is a positive thing for COVID because it is an enveloped virus. And this is one of the reasons that the use of the hand sanitizer and all of that is important, because it actually is effective against it. All right, so let's look at our terms for this chapter and start to understand what the difference is between these different ones. This again is a chart table that I took from the OpenStax link that I gave you for that microbiology text, but I really like the way that it summarizes the difference because I think it breaks it down to really simple. So remember, fomites are inanimate objects. So you'll see in all of these, it says inanimate, for all three of these. So disinfection, sanitization, and sterilization are all for use on inanimate objects, whereas the ones on the bottom here are for use on living tissue. So disinfection, if you see, reduces the microbial load. That means that it doesn't get rid of all of them. It just reduces the load to an acceptable level. So disinfection is not the same as sterilization. Sterilization is completely eliminating all vegetative cells, endospores, and viruses. And then this chart gives you common applications and common agents for each one of these. Sanitization, we were talking about earlier, is for use on eating utensils and things like public restrooms. So commercial dishwashing is an example of sanitization, cleaning eating utensils and things like that. And then on living tissue, you have antisepsis and degerming. So antisepsis would be things like using isopropyl alcohol, iodine, hydrogen peroxide. So these are cleaning the skin. So again, these are for use on living tissues. And then degerming is reducing the microbial load by gentle scrubbing, or can also be the use of an alcohol swab to reduce it. So this would be hand washing or cleaning an injection site, for example. All right, issues to consider when killing an organism. We went through this morning in the tutoring and study guide review, but this is one of the items that you needed to know for the study guide. So this is knowing the things that are important in order to determine what's the correct method to use for antimicrobial action. So number and type of organism. Type of organism obviously is a huge issue. Type of agent for the infection control, strength of the killing agent, amount of exposure time required, and then environmental issues such as temperature can also be important. So type of compound that you're treating is also important as to whether it's sensitive to the agent that you're using to clean it. Methods of use for disinfection and sterilization. These are the different ones that we're going to kind of walk through, and you just need to understand the basic parameters of these. And some of these overlap. So you're going to have heat, but you also have heat that's involved in pasteurization, boiling water, steam, and pressure. So there's just different ways to apply these. So heat is commonly used for disinfecting and sterilizing. This denatures proteins that is going to cause the cells to coagulate, kind of clump together and die. Dry heat is for objects that could be damaged by moist heat. A lot of times moist heat is more effective. With steam, you can get it hotter. But dry heat can be used often for glass, for example. Moist heat, like I said, is more effective, and the autoclave is pressurized moist heat. So you definitely need to know that the autoclave is the gold standard for sterilization. And then incineration would be basically heating something to ashes. So it's completely destroyed, disintegrated. Obviously, incineration can't be used on something that you need to use afterwards. So that's how the autoclave is very helpful. Heated water kills vegetative cells and denatures most viruses. If somebody's using this on an instrument, it needs to be immersed in 70 degrees water for at least 30 minutes and then dried and packaged in a sterile manner. Boiling water kills most bacteria and viruses after 15 minutes, but it's not considered a true sterilization process. So this may be effective against fungal spores, but it may not be effective against bacterial endospores. And then if it's in a higher altitude for every 1,000 feet, it needs to increase the time by 5 minutes. Steam and pressure. I had to add that one in the other one also, because you definitely need to know the autoclave. But the most effective sterilization method is the autoclave. You are actually, oh, I almost forgot to mention this. Your week 4, well, now you don't have to rush because you're not taking the midterm until week 5, but your week 4 lab is on pasteurization and sterilization. And it's really, really going to be helpful for the material that's in this lecture to help you learn it and understand it and everything. So make sure that you complete that lab before week 5 so that you have that information to help you for the midterm. All right, for the autoclave, there's a heat sensitive indicator that is usually on the package that you have the items in the autoclave, and it changes color once the autoclave has gotten to the temperature and pressure that makes it effective. So you can actually see the indicator change when you take it out to verify that it worked. But this kills bacteria, fungi, and spores and denatures viruses. Doesn't say prions on there, so you're not guaranteed that it's going to destroy prions. Disadvantage of the autoclave, like I said, is that it can't be used on heat labile, meaning heat sensitive materials. I'm typing that in there just because in our other curriculum, the heat labile term came up and it really threw students off. So I want to make sure everybody realizes that heat labile means that it cannot withstand heat. It cannot be disinfected or sterilized by a method that uses heat. All right, liquids and compounds. This includes all of these different types that you're going to kind of walk through. These are the most common types that you might actually come into contact with. So these are probably the ones that you should know. There's a chart at a table at the end of this that kind of summarizes them a little bit better. But alcohol disorganizes lipid structures of the cell and denatures proteins. This is effective against gram-positive, gram-negative, and acid-fast bacteria. It is not effective against spores. And if it's too concentrated, it can irritate the skin. So it depends on what percentage concentration you're using for this. It may also damage plastic or rubber. This can be in the form of ethanol, which is effective at 70% concentration. If you've seen any of the news stories or cautions about the hand sanitizers, they say that it needs to be at least 70% concentration in order for it to be effective. So if you ever see one that's below that, it's not going to be as effective. And then isopropyl alcohol is most effective at a 90% concentration. Acetic acid is vinegar. This is used as a food preservative and inhibits the growth of many bacteria and fungi. So it is not a sterilization, it just inhibits the growth. The acidity denatures the cell's proteins, and white distilled vinegar is the preferred type. And then phenols, this is the example that we talked about when we talked about Lister learning about or discovering aseptic technique, where he used phenols on the instruments before surgery and discovered that they decreased the amount of infections that were occurring. These cause cell leakage and inactivate enzymes in the cell membrane. It does inactivate viruses. I almost said killed, but we don't say killed viruses since they're not living. So we'll say inactivates viruses, virucidal. It is not sporicidal, so it does not destroy spores. Most commonly used in cleaning instruments and general housekeeping, but it is corrosive to the skin, so it should not be used in contact with skin. All right, chlorine can be in gas or liquid form. It's effective against most bacteria, viruses, and fungi. Again, it is not sporicidal, at least not at room temperature. It can be highly corrosive. We probably all know that, but it can be highly corrosive to several things. And obviously you don't want that to come into contact with your skin either. And then another version is hexachlorophene, which is the disinfectant without the chlorine smell. Depending on the purpose of it, it can be used at different concentrations. So 1 to 10 dilution is recommended for cleaning blood spills. For example, a 1 to 50 solution is effective against gram-negative bacteria, spores, Mycobacterium tuberculosis with a 10-minute exposure time. I think we used 10% bleach to clean the benches when we were using it in the actual microbiology lab at AZC. Hydrogen peroxide can be used in a 3% solution or a 6% solution. It's generally not considered one of the best for wound cleaning, but it is used often. So this can be a mild antiseptic for wound cleaning. And it can cause damage to a wound tissue at a greater than 3%. But at 6%, it's considered bactericidal, virucidal, fungicidal with a 10-minute exposure time at room temperature, which is probably longer than we would generally just put it on if you had like a scratch or something like that. Sporicidal after a 6-hour exposure. So a long time. And then quats are the abbreviation you may have heard if you've heard of quats, quaternary ammonium compounds. These cause loss of semipermeability in the cell membrane that leads to lysis. And these are bactericidal, especially gram-positive. We all know gram-positive are more susceptible to antimicrobials than gram-negative. It is not effective against bacterial spores, enteroviruses, hepatitis B, some fungi, and spores that cause tuberculosis. I find that interesting because we used quats as the cleaning agent in one of the other microbiology labs that I worked with, and we worked with all different kinds of bacteria. So I'm surprised that that was their choice, since it seems like it's not as effective. Glutaraldehydes used for disinfecting, sterilizing surgical instruments and other medical equipment. It's bactericidal, tuberculocidal, fungicidal, virucidal with 10 to 30-minute exposure. Can be sporicidal with a 10-hour exposure time. Interrupts metabolism and reproduction in the organisms. And then glutaraldehydes can also irritate the skin and mucous membranes, can cause damage to rubber and plastics and steel, and then also requires rinsing and drying and proper packaging of items after being soaked. We already looked at the zone of inhibitions before, but you can use these 4 antibiotics to determine susceptibility to antibiotic treatment, as well as the different antimicrobial agents that are chemical compounds. So this is just an example showing you, for example, we'll look at the E. coli, and then we'll stop after that. We'll pick up on next week. E. coli is the one that is most susceptible to the effective most against E. coli would be chlorine, because you see this large zone of inhibition. And then your alpha phenol is a little effective, but not as effective as chlorine. And then same with quat. And then here you see hexachlorophene is not effective at all against E. coli because there is no zone of inhibition. So just in case you didn't understand that whenever we went through it before, there's another example kind of showing you the difference. And you can see the susceptibility of different organisms to these different compounds can vary quite a bit. And so this is why these types of tests are helpful to determine susceptibility. And then this is that chart that I mentioned that is a little bit more simple than the other charts that were in here, that shows the most resistant to antimicrobials at the top is the prions, like we were talking about before, endospores that you probably gathered. And then it just progresses all the way down to envelope viruses being the most susceptible. All right, so I'm going to stop here. And then, like I said, I will give you an update if I hear anything else about the midterm and whether we need to cover more material and whatnot. Other than that, I hope everybody has a wonderful weekend. And like I said, just reach out if you have any questions that I can't answer.
Thank you.
Thank you. Good night.
Thank you.